Hi, good morning. My name is, as Valentina nicely introduced me, Gryte Satas. I'm a grad student in Ben Raphael's lab at Princeton University right now. I'm going to talk to you about some new approaches towards inferring tumor phylogenies from cancer sequencing data. So we can think of cancer as an evolutionary process, and this is characterized by the accumulation of mutations in a population of cells over time. As some of these cells may confer a selective advantage to the subset of cells that some of these mutations may confer a selective advantage to the subset of cells that contain them, we find that oftentimes this process ends up with a tumor that is actually highly heterogeneous, with populations of cells that contain distinct sets of somatic mutations. And understanding this heterogeneity and evolutionary process is important for improving our treatment of cancer, as heterogeneity has been associated with increased resistance to treatment, more aggressive course of disease, as well as an increased likelihood of relapse and metastasis. And being able to accurately characterize this process of tumor evolution can really help us recognize common patterns, as well as understanding metastatic development, which has been associated with up to 90% of deaths from solid tumors, as well as identifying potential targets for treatment for individual patients. So as any evolutionary process, we can model cancer evolution as a phylogenetic tree, where the nodes here correspond to clones, which are collections of cells that have the same set of somatic mutations. And the edges correspond to direct ancestral relationships between these clones. So typically in a phylogenetic study, we measure individuals from a population, and we measure some set of features about them. These could be physical or genetic features. And we use these features to reconstruct the evolutionary history going backwards in time. There's a rich body of work describing how to go from these features to a phylogenetic tree. When we talk about cancer sequencing, the picture gets a little bit more complicated. Because the vast majority of cancer sequencing that's done nowadays is bulk sequencing, where instead of measuring individual cells, we're actually taking tens of thousands of cells in the same sample. And so the data that we observe is actually representative of the mixture of the genomic content of all of these cells together. And so we can think of each of these samples as being a mixture of leaves from our phylogeny, where the leaves are the clones that are present at time of biopsy. Taking a closer look at what the data we actually observe looks like, I'm going to talk today about single nucleotide variants, and what we actually observe for each variant is a frequency, and we're going to define this as the proportion of reads that align to that location that contain the variant. And we make the assumption that this proportion is proportional to the percent of cells in the original mixture that have the mutation. And this assumption will generally hold as long as that variant is not affected by copy number aberrations that change the number of mutations that each cell has, and we can generally measure this. So the fundamental problem that we're looking at today is what I call the Tumor Phylogeny Inference problem, is given these frequencies, how do we reconstruct the original phylogenetic tree that generated the frequencies? And this problem is a little bit underspecified, that we really need to define an evolutionary model that we're considering, and there's any number of models that we talk about: maximum likelihood, maximum parsimony, and for today I'm going to look at what's possibly the simplest model, is the infinite sites model, which assumes that a mutation is going to occur at most once in the lifetime of a tumor, and it will never subsequently be lost. This model has been widely used for single nucleotide variants in cancer, as we have a relatively short evolutionary time span and relatively few mutations compared to the length of the genome. What we really want to look at is how the phylogeny actually constrains the set of frequencies that it's possible to observe in order to be able to reconstruct the phylogeny from the observed frequencies. So I'm going to start by talking about some past work that's described a structure to solutions of this problem. So we're going to look at one sample to begin with. And suppose we observe some set of mutation frequencies for this sample. The constraint that the tree actually places on these observed frequencies is what's been called the sum condition. It just states that for any node in the tree, the frequency of the mutations of that node have to be greater than or equal to the sum of the frequencies of all the nodes, of all the mutations in children nodes, and in direct child nodes, rather. And this constraint really makes sense if you think about it, as since we know we never lose mutations under the infinite sites model, any descendant clones must contain the original mutation. And we can extend this to multiple samples by just stating it has to hold across all samples. This paper from El-Kabir et al. From 2015 proved that this is really a necessary and sufficient condition for solutions, that frequencies admit trees if and only if this sum condition actually holds for F and T. And went on to characterize solutions to our tumor phylogeny inference problem. There's trees that respect the sum condition for a set of frequencies as being a set of constrained spanning trees from a particular graph, where the constraint is actually just this sum condition. So there's been a number of methods that have used combinatorial constraints to reconstruct phylogenies from frequencies using both the infinite sites model and a number of other models, evolutionary models. And really, if the frequencies were exact measures of the underlying frequencies and our data were perfect and noise-free, this would be the end of the story, and my talk would have been very short. But as we all know, real data is a little bit messier, that we have sequencing noise in our measured data. And so the question is, how do we handle this sequencing noise as the relationship between the tree and frequencies is a little bit fuzzier in this case. The natural approach is to apply a probabilistic model for our sequencing data. And we find that when we do this, we often end up with mutations that have indistinguishable frequencies, that we can't actually tell which one has a higher underlying frequency and which one occurred earlier in evolutionary time than others. And moreover, as most mutations in the evolutionary history of a cancer are neutral, do not confer a selective advantage, we find that they tend to cluster on the tree and as a result have frequencies that are similar. So the approach that a lot of people have taken is to cluster mutations together prior to building a tree. And so there's been a number of methods specifically designed to cluster observed sequencing, to cluster mutations given the observed frequencies. So these methods do not have a notion of an underlying tree generating this data. And so what we find is that oftentimes the frequencies that we actually get do not admit any tree. They're somehow contradictory and do not respect our constraints. So the natural next step to take would be to try to do both of these steps together. So if while we're clustering, we can enforce that our frequencies have to respect a tree constraint, then we're guaranteed to get a tree in the end. There's been a number of more recent methods that try to do this sort of tree-structured clustering while simultaneously inferring a phylogeny. And they generally take a sampling approach of Markov chain Monte Carlo, where they're sampling from the space of trees, frequencies, and cluster assignment for our mutations. And this is a highly complex, highly dimensional space to sample from. And while MCMC approaches have guarantees of convergence in the limit to infinity, they make no guarantees about actually converging in a reasonable amount of time that we have. So what we found is that often running these methods, that while they have a reasonable generative model for the data, they don't necessarily converge to the maximum likelihood solution in a time that's really practical for people trying to use this to actually get phylogenies from real data. Which brings me to the new work that I'm talking about today, which is combines some of the ideas that I already presented of a probabilistic and combinatorial approach towards inferring tumor phylogenies. So the quantity of interest that we're going to look at is the posterior likelihood of a tree given our data. We can note that our observed data, the allele frequencies, is actually independent of the tree, conditionally independent of the tree, given the underlying cluster frequencies. That is, the sequencing noise that we observe, the sequencing frequencies that we observe, comes only from the underlying mixture proportions in the sample that we took, and knowing that they're independent of the actual underlying tree. So we can use this idea to decompose our posterior likelihood into two terms. The first one is the probability of our data given the frequencies, which is just a model for sequencing noise. And the second one being the probability of our frequencies given the tree. And there's a number of ways that we can model this term. For now, I'm going to stick to the simplest possible one, which is a binary indicator, which says that if a set of frequencies respects our tree constraint, then it has uniform probability, and if a set of frequencies violates our tree constraint, then it's not allowed. It has probability 0. So the way to really read this is that the highest probability trees, the trees that we're interested in, are those which admit frequencies that have high data likelihood. So stepping back to look at this at a conceptual level, we have some complex probability space over our set of frequencies, and I'm showing it a visualization of it in 2 dimensions. In reality, it's highly dimensional. And we're decomposing this into a likelihood function over our data and a set of combinatorial constraints. And to calculate the integral, we're really going to take advantage of both of these parts and the fact that we can accurately characterize both of these parts. So we're going to use our data likelihood to focus sampling onto areas of high probability. And then we're going to use the combinatorial constraints to prune our computation. So that we don't have to consider samples that have 0 probability. So the statistical technique that we use for this is called importance sampling. And generally speaking, importance sampling, one of the uses for it is Monte Carlo integration. It's when we have some distribution that we can evaluate. But if we're trying to integrate, it's hard to integrate analytically, and it's hard to sample from. So what we can do is find a distribution that approximates our distribution of interest, and it's easy to sample from. And then we could alternatively sample from this proposal distribution. And then we can correct our samples by the probability of sampling it from the proposal distribution. And this will actually converge to our integral of interest. So we combine these ideas into an algorithm that we call pastry. And pastry, we take in our given allele frequency data. And for our proposal distribution, we're going to use one of these unstructured clustering approaches to give us a proposed clustering, something that if we ignore the tree constraint looks pretty good. We calculate a posterior distribution over the cluster frequencies. And then we sample from this posterior distribution. For each sample, we enumerate the set of constrained spanning trees. And since these trees are the only ones for which the sample would actually have a probability that's non-zero, we contribute that sample to only those trees. And so we're able to calculate the posterior likelihood over the trees in this way. I'm going to show some results comparing this to existing algorithms for doing this, including Ancestry, which is a combinatorial approach, and Phylosub and Canopy, which are our probabilistic approaches, these MCMC approaches. And we find that in terms of tree reconstruction, pastry is able to outperform all of these. And moreover, it's able to really improve on the runtime of some of these probabilistic algorithms. I'll note that this is a log scale. So really we're able to run on the order of minutes, while some of these other approaches took hours. Ancestry is still a little bit faster, which makes sense since we're using the same combinatorial structure, but we're taking this iterative approach. But we are still able to get a performance improvement over them. And sorry, having clicker difficulties. There we go. So we applied this to some real data, leukemia. We used a cohort of patients from a study from leukemia last year, where they took longitudinal data from leukemia patients of pretreatment, post-treatment, and then possibly 1 or 2 follow-up studies, and classified these into linear and branched evolution. And they noted that branched evolution is really more frequent in cases with progressive disease. And so we took a look at one patient, which had three samples, and was originally classified as having a complex branch phylogeny by Phylosub, which is one of these probabilistic approaches. And we found that running our approach on it, we get something that's actually rather linear, where the optimal tree is mostly linear, and the fully linear tree is a third out of 115 possible tree topologies. And moreover, the data likelihood that we get under these trees is actually significantly better than the data likelihood under Phylosub. And just to make sure that this wasn't a difference in how we're modeling sequencing data, as Phylosub has a slightly different data model. Sorry. There we go. We recalculated the data likelihood under their model and found that even here, we find a better likelihood. And this is really indicative of the fact that in the time allotted, Phylosub was not able to converge to the optimal solution. In the interest of time, I'm going to skip through those slides, but you're welcome to ask me about them. In conclusion, today I talked to you about really the importance of characterizing tumor evolution and having proper approaches towards being able to infer phylogenies from our complex bulk sequencing data. And really, I guess, the main takeaway that I want to emphasize from this talk to people that might not necessarily be involved in cancer is that if we know a little bit about the combinatorial structure that underlies our problem, we can combine it with some of these statistical techniques to really be able to improve the efficiency and accuracy of inference. And finally, I presented a new method that's able to really infer tumor phylogenies more quickly and accurately from bulk tumor samples. I'd like to acknowledge my whole group, especially my advisor, Ben Raphael, who's also my co-author on this work. And I'd like to thank HiTSeq for inviting me to speak and putting together such an exciting lineup today and tomorrow, as well as providing travel funding.
Questions? Okay.
Thank you for the talk. One very small, perhaps naive question. Did you evaluate the performance of your method with varying depth of sequencing? I saw your simulated data as about 200x. So that's why I was wondering.
Yeah.
Okay, you see.
Yeah. So we did take a look at what we get with higher coverage, and we do find that as coverage increases, we get significantly improved performance. The plot that I don't have on here is actually with number of samples, and the interesting result that we get is actually as we improve samples, we get a far greater increase in performance even than we see with coverage. So yeah, at some of these lower depths, it's a little bit more difficult to have certainty in ancestral relationships.
Okay, thank you very much.
Great talk, thank you. So one thing we see in prostate cancer is that in about 15% of cases, you'll actually see multiple primaries, so the same patient having multiple tumors in one prostate. Does your tool ever output multiple trees to show that, or do you always converge to one tree?
So generally speaking, well, I guess what multiple primaries would look like in this context would be that we have a single tree, but we have nodes at the top that don't necessarily have somatic mutations, because these cells obviously do have a common ancestor being from the same patient. So it is possible. I don't think we've ever actually observed that, though. And perhaps applying it to some of these prostate cancer data sets would be interesting for that.
All right, cool, thanks.
Could you Gryte again?
Okay.
So now we welcome